So I'm really enjoying today's topics because we've gone from Mike talking big picture benefits of cleaning, and then Jean talking about methodologies and measurements of methodologies. And now we've got into this, I love this, real application of how you can do this kind of testing. And I'm going to talk to you about a simple test, one of those sort of things that Jean mentioned that we can do in colleges with the use of the cheap student labor. A simple test that we developed to look at different mopping technologies. And a client came to us a few years ago and asked us if we could develop a technology that could compare different kinds of mopping. And just to tell you, in our center at Simmons, we were able not just to look, I'm just going to talk about a small part of this project, which was the microbiology of mopping, but we actually were also able to do, go into people's homes and evaluate the product's use in the home, evaluate consumer practices around mopping. We were able to do an ergonomic evaluation of mopping, and as well as an economic evaluation. So those are all the kinds of things that we can do in this sort of academic college environment. And what's very interesting is we're trying to, in this project, we were trying to develop a test in the laboratory that wasn't just a lab test that really mimicked real-life conditions, but we developed it in the lab, and we called it an in-use laboratory test, so that we could really control for confounding factors and, come up with some sort of statistically valid data. It's also something that we haven't mentioned is the speed at which cleaning products, cleaning technologies are changing, and how we don't always catch up with those changes and don't always have the possibility to evaluate them. One really good example that's written about in the literature is how over the last decade or so, people's consumer practices around laundering have changed. And it's interesting to note that the practices have changed for a number of different reasons. One is that because of economy, people are using cold water washing now. Does that have an impact on the hygiene practices of laundry? Companies have removed, because of sort of environmental concerns, chlorine and phosphates from laundry powders. Does that have an impact on laundry hygiene? And so it wasn't until someone said, hey, let's take a look at this. Are we doing something differently in laundry now, that we actually were able to determine that, in fact, yes, cold water washing isn't necessarily a hygienic process anymore. It doesn't necessarily remove pathogens from the laundry process, and there are bacteria and viruses that can survive cold water washing. So I'm just trying to make the point that changes happen for various reasons, and then we kind of have to try and catch up with them and understand what's going on. So there are a lot of, in the consumer market at least, there are new and improved cleaning devices out there which offer benefits to the consumer in terms of convenience, cost, efficiency. They offer, or they claim to offer, improved cleaning power, and some of them offer removal of microbes. And again, it's not until you actually go out and sort of try and test these claims and test these technologies that you really begin to understand what's going on. And so consumer mopping technology is a technology that is changing quite rapidly. There is the introduction of many disposable one-use type mop appliances into the consumer market that are becoming very popular. They're very convenient, they're very easy to use, and they seem to be very well received. So our job was to sort of look at mopping and try and determine, to try and break it down. Mopping sounds like it's such a mundane procedure. That's so easy. It would be so easy to analyze mopping practice. But when you take any one of these practices and you have to deconstruct it and break it down into its component parts, which is what you have to do to develop a sort of standard in-use test methodology. Even the most mundane practice, such as mopping, can become quite complex to work with. So the first thing we had to do was sort of try and think, okay, in a mopping procedure, where might the risks of, and I was looking at it from a hygiene, bacterial cross-contamination point of view. So in a mopping practice, where might there be any risks of cross-contamination associated with mopping? So we predicted that there would be three things that we probably should be looking at. One of which was the potential of a mop to transfer bacteria from one floor surface to another. That was number one. Number two is the potential for transfer of contamination from the mop to the hands of the operator, just by handling the mop and whatever one does with mops. And the third area was for mopping practices that are associated with a bucket and a bucket of mopping fluid, the potential for contamination, cross-contamination from that mopping fluid as it's disposed of. And for consumers, the most common place that they're going to dispose of bucket mopping fluid is the kitchen sink. So this potential of transferring contamination from a floor now into a kitchen sink area. And by the way, you know, consumer kitchens are a very interesting place to do these kind of studies because a consumer kitchen, it kind of covers all evils, everything. Everything happens in the kitchen. You know, the kitchen is the place you're supposed to be preparing food, and if you went into a commercial kitchen or the kitchen next door here, hopefully all we'd see them doing was preparing food. But in a consumer kitchen, we have food preparation, we have mopping fluids poured down the kitchen sink. Often there's laundry practices going on in the consumer kitchen. It's where the pets get fed. It's where you clean out the pet mouse's cage. It's in the kitchen sink. You clean, you know, the potting of the plants. Everything's going on there in the consumer kitchen. So it's quite a lab, a laboratory experience, just working in a consumer kitchen and seeing what's going on. Anyway, our challenge was to break down this process and develop an in-use test that we could get some data from, that we could actually do some statistical comparisons. So the challenge, I think I've already mentioned, and the criteria for evaluation that we were using, we were using biomarkers, we were using bacterial cross-contamination to evaluate our different test methods. Another question when you're developing these kind of tests is, well, what would you use for bacteria in this test? You know, if you just use the bacteria that are on the floor anyway, well, that's kind of pretty uncontrolled. You'd have, if you just went and mopped, if I went into different kitchens and mopped different floors, I wouldn't know what kind of bacteria were there to start with. They would be different numbers. So we came up with this idea of actually extracting an inoculum of bacteria from a mop that had previously been in use. And that's what we did. And we used that inoculum of bacteria for all our tests, so it became our standardized test inoculum. We deconstructed the mopping practice and broke it down into its component parts and standardized them, and so that we could repeat them. Every time we did the test in a very standard method. And we also built our own little model floors on which to do this testing. So I think I've mentioned most of these. So our test was composed of model floors. It was composed of a wild-type bacterial inoculum that we'd harvested, and we froze it down, and we used it each time we did the test. We had several different mops in this test that we compared. We standardized the mopping procedure, and we used standard bacterial sampling techniques and counting techniques to be able to compare the different mopping procedures that we used. So here were our little model floors. My students and I had to run to Home Depot and get all these bits and pieces and get into floor construction and carpentry. So we built these 2-by-4-foot sections of floor, and we glued on vinyl-covered flooring onto these floors. And then we marked out 3 areas. We permanently marked these floors with 3 areas that we were going to test. So in each test, we tested the same area on the floors. We also marked out an area where we would put down, so we were going to actually inoculate this floor and mop it and then test it and see what bacteria we would get back following mopping. And so we also had a control area where we put down our test inoculum, and we didn't mop that, and we just sampled it at the end just to see how many bacteria we had left on the floor, without doing anything to it. We also had an area that we did some air sampling. This picture actually shows us just using a Petri dish, an open Petri dish for just catching fallout. We were interested to know if there was any bacteria falling out from the air, into the air, and from the air onto the floors from the mopping process. But we also later employed a biotest Hycon air sampler, which we would run whilst we were mopping. So we had this air sampler sitting here that would be sucking in air whilst we were mopping, and then the air would be collected onto a strip of agar in the little test appliance, and we could look at the counts that way. We pre-disinfected, every time we did a test, we pre-disinfected absolutely everything involved with 70% ethanol, so that, with alcohol, so that there was no pre-contamination, so that everything that we were following was that we applied to the test and we took back from the test. And we also regularly tested to make sure that we really didn't have any other contamination. So we would do just general sampling to make sure that our ethanol was doing the job that we wanted it to do. So harvesting the inoculum was pretty innovative. This work was actually done at the Procter & Gamble Home Care Research Lab in Cincinnati, when we took a string mop that had been in use for a year in domestic use, and we put it in the mop head into a sterile plastic bag, and we massaged it in phosphate buffered fluid in this bag. Pretty weird procedure. And then we collected the fluid that from the bag, and we centrifuged it down into 20 ml aliquots for 15 minutes. And then we took the pellets that were centrifuged, and we resuspend them in a medium, a cryogenic medium, that allows you to freeze down the bacteria then and keep them viable whilst they're frozen. So we dispensed this into 2 ml aliquots that we froze down and transported to our lab in Boston. So we had a whole range of samples that were all exactly the same, little vials that we could use every time we did the test. We would open up a new vial, take it out the freezer, unfreeze it, and use it. So we analyzed our inoculum, our wild harvest inoculum, and we found that it contained about 80 million organisms, 80 million bacteria per milliliter of fluid. This was the stuff we had collected from a mop that had been in use. And we did some identification work by standard biochemical testing and by genomic analysis, RNA analysis, and we found that our inoculum included a range of bacteria that kind of didn't surprise us, that these were the bacteria that we would find. These are the Micrococcus, Pseudomonas, Stenotrophomonas, are pretty common organisms in the home environment. We know from other work that we've done in the home that we do find these organisms in the general environment. What did surprise us was we didn't really find any gram-negative bacteria in our one-year-old mop that had been, that was just generally being used. But this was the inoculum we got. This was the test inoculum that we used every time we did a test. And so the way that we would conduct the test, that we would apply 1 milliliter of this inoculum to field 1. Which way is it? to here, to this field, and then we would spread it with a glass spreader onto that field, and then we would allow it to dry down for 45 minutes before we actually carried out any mopping practice. And it's interesting to note that just drying itself, and this goes back to something I mentioned yesterday in terms of disinfection procedures, drying itself is a disinfection procedure. And you can see that the inoculum dropped 3 logs. We got a 3-log reduction just by drying the bacteria onto the surface. So it was a 99.9% reduction in bacterial count just drying it onto the surface. So we were left with about 60,000 bacteria on our surface to work with for mopping. And again, we know from studies that we've done in kitchens, sampling that we've done in homes, that that's a kind of pretty conservative number of bacteria to find on a kitchen floor. We wouldn't be surprised to find up to 1 million bacteria per sample area on a kitchen floor. So it's not like we weren't overloading this. We weren't sort of putting so many bacteria onto the floor that we were sort of biasing our experiment. So we used, for our mopping mops, we used sponge and string mops that had been in use, and we had claimed them back from householders and put them into our test just as they were. We used new per-use sponge and string mop heads, so every time we did a test we would put a new mop head on. And we used a generic floor detergent that we made up according to the label instructions. It was not an antibacterial or antimicrobial detergent. And we also used this disposable pre-moistened mop, this Swiffer mop, with a pre-moistened wet cloth that comes with these mops. So those were the 3 the different mops that we used. So in total we used 5 different mopping procedures. Use string, use sponge, clean string, clean sponge, and then this disposable mop. And we did each trial 10 times, so that we had a total of 50 trials in total to work with. So our sponge and string mops, we wetted them in the bucket solution. We placed all the mops on the floor so that they were in contact with the coving. The edge of the mop was sitting on the floor, but it was also in contact with the coving at the side of the floor. And then we pushed the mops across the floor through our 3 test fields, and then we pulled them back through the test fields again. And any bucket fluid was always disposed. We had a test sink in our lab, and we disposed of everything into that test sink. The operator used gloved hands for carrying out the procedure, but when the operator then had to dispose of the mop fluid, she would take off her gloves in order to do that part of the practice, so that she would come into contact with mopping fluid and with mop heads. And she also would contact the faucet of the sink, just to try and mimic, sort of what's happening in real life in someone's kitchen. So we sampled 3 different ways, depending on the kind of surface that we were sampling from. We used a swabbing technique, the settle plate that I showed you, or the Biocon air tester. And for hands, we contacted them directly onto agar. So you just put a person's hand directly down onto an agar plate, and you get a sort of mirror image of the bacteria that will come off the hand and sit and grow up on the agar plate. It's a standard bacteriological lab procedure. It's called a serial dilution. We diluted our samples so that we would be able to count the bacteria that were in the samples, and we incubated for 24 hours. So our results, just to briefly show you the kind of results that we were getting. This table shows you the average bacterial counts from 10 trials for each of the mop systems that we used. So we used, as I mentioned, used sponge mop, new sponge mop, used string, new string, and the disposable mop at the end. And then down here you have the different surfaces that we sampled. And here you can see the range of average counts that we got. So if we look at floor fields, the first field that we sampled, 3 fields that we sampled on the floor, you can see that for used mops, the count actually increased on the floor after mopping. Remember, we were starting with a viable count of bacteria of about 10 to the 4 on the floor, and count actually increased, which tells you that the mop was actually adding to the contamination. The used mops were adding to the contamination levels on the floor. Whereas when we used new mop heads and the disposable mop, we got reductions in the order of up to 10 to the 2, log reductions of between 1 and 2 log reductions. So we were getting a 90 to 99% reduction of counts on the floor. Interesting to note that we had cross-contamination to the coving. We never applied bacteria to the coving, but just being in contact with the mops. I don't know that there's any particular significance in coving becoming contaminated, but this was just a surface that was in contact with the mops and became contaminated. The sinks. There's no data for the disposable mop because, of course, this is a bucketless, waterless system, so we didn't have to dispose of any fluid with this disposable mop system. But you can see that in every case, there was contamination transferred of the traditional mops. There was contamination transferred to the sink, the bottom of the sink, the surrounding areas of the sink, to the tap, and of course there was contamination in the bucket. Not a big surprise. This didn't surprise us because, if you're collecting bacteria off a floor and you're putting them into fluid in a bucket and then you dispose of the bucket into the sink, we're not surprised to see that you're actually transferring cross-contaminating bacteria into the sink. But it just does, again, illustrate that these sort of fluids, dirty cleaning fluids, if they're not disposed of carefully, and clearly the kitchen sink isn't the right place to dispose of this kind of fluids, they are causing cross-contamination. You also see counts on the hands. You see a maximum of 250, or more than 250. 250 was our maximum counting capability for that direct hand contact technique. So that's a real limitation in that technique, that we couldn't count anything more than 250. But it's quite likely there was much higher levels of contamination on the hands, much less contamination associated with the disposable mop, because there's much less handling. The operator just had to grab the edge of that disposable cloth and put it in the trash, so you're not in contact with contaminated fluids. These results are easier to see if you illustrate them, and we gave the data to our statistician, and he did a statistical testing comparing the population medians and means for these results. And what these two plots really show you is that here are the used sponge and used string mops. You're getting counts in the region of 100,000 on the floor after you've used these mops, whereas down here you have counts in the regions of 1,000 to 5,000 after using clean mop heads and disposable mops. And you can see there's no overlap here. They are statistically very different. And there's very high confidence. There's a 95% confidence interval, meaning that we are 95% confident that these averages are where they appear to be, that they're accurate. Same thing for the coving sites. Again, much more contamination transferred to another site. using the used mops than the clean or the disposable mops. And again, statistically significant data there. So what were the limitations, or what are the limitations to this kind of testing? And this kind of testing is like the next stage on from the testing we were talking about yesterday, where you do a first test in a laboratory, and then you do a second test, which is an in-use test, which is trying to mimic what's happening in real life. And then the final stage would be actually to go out in the field and test. So the limitations on these kind of tests are that they are complex to construct and perform. They're not necessarily expensive, but they're rather lengthy to understand what's going on. And then, of course, we were using such a small-scale model. You know, how do you relate that model to real life? How do you ramp up into real life? And would the outcomes be similar in a real-life situation when you're covering a much larger floor area than we were covering? So when you interpret these kind of results, these results would indicate that, for example, this disposable mop was a very effective means of reducing the potential for cross-contamination. We would need to go out and test that in a real-life situation to know if that was really going to translate. So you have to interpret the data within the framework and context of the test procedure. Our inoculum didn't contain pathogens that we might expect to find on floors. As I said, we didn't find any gram-negative bacteria, and so that's another limitation. We're not sure from this test how they would perform. And we didn't use any disinfectants in this test. We only tested with a detergent. So we would expect to see different results, but we could definitely test that, different results, when you used an effective disinfectant system. So in summary, we would say that you can use this kind of modeling to evaluate the hygienic outcomes from the different mopping procedures. We know that these tests are complex to develop. We know that we can test in such a way that we can get data that we can do statistical analysis on, so we can make statistical comparisons between different test systems. We knew already, but we confirmed again that mopping can result in cross-contamination across floors if it's not carried out in a correct manner. We showed again that using old equipment can cause additional problems, can add contamination to a cleaning procedure rather than remove contamination. We confirmed again that the dirty mopping fluid is a source of cross-contamination to hands and to wherever it's disposed of, so better dispose of it in the right place. And we could conclude that at least in the test model that we used, in the confines and context of this test, that this waterless disposable mopping system did actually reduce the risk of cross-contamination. And I'd just like to thank the people who worked in our lab on this test.